Good afternoon, and welcome to the Akoya Biosciences User Group Meeting webinar series. My name is Daniel Vaughan, and I will be your host. Our goal of the meeting today is to share the advances we are making in spatial biology. The webinar is intended to feel like a symposium, sort of focused on the science and the research within this area. We want to create a connection between Akoya and yourselves, and also try and provide a platform for you to share your valuable insights with each other. This is the second of the 3 meetings planned. We had our first webinar last week, and our final of the series will be next week. You can find the recordings of these webinars on the Akoya Biosciences website. The focus of today's user meeting is how Akoya Biosciences technology is being utilized in biomarker research. At this point, I would like to thank all of the speakers in advance for taking the time out of their busy schedules. I'm sure we all appreciate what is required to prepare for such presentations. To enable us to stick to the allocated time, we will cover the questions at the end once all of the presentations are complete. If we don't have time to respond to all of the questions in the allocated time, our application specialist will follow up with you directly. Myself and Virginie Goubert, one of our application scientists, will moderate the questions to the speakers at the end. And of course, if there is a technical element outside of the speaker's expertise, one of the application specialists will step in to respond. So our 3 guest speakers today join us from the UK, Spain, and Germany. And we also have one of our senior application specialists to kick off the meeting with the first talk. So at this point, it gives me great pleasure to introduce my colleague, Dr. Bjorn Vendick. Bjorn is by background is a developmental biologist. During his early career, he worked in an imaging facility. Bjorn's commercial experience includes working as a senior confocal specialist, and now as one of the Akoya team, he is one of our imaging specialists covering the whole workflow from staining to image analysis.
Okay, welcome to our second user group meeting session here. And I'm very happy that we could have this year a user group meeting, which wasn't so clear. Now we have to do remotely, but I think everybody's already used to having remote sessions, and it also has quite some advantages, as you know, and I'm happy to start off this session here. What I would like to give you is a little bit an overview of our main field, which is immuno-oncology, but also going way beyond this. And I would like to show you a few publications that came out in the last couple of years, but also to show you some applications where our technology is used and how far you can get with this technology. And this should include a little bit showing how people use our technology in a fully automated workflow. I will also show you that it's used in vaccination application, also pancreatic cancer, which is a very complicated cancer to work on. I will show you some advantages, how our technology is used for skin inflammation in mouse model. And also, we have now a strong emerging field in transplantation and also in neuroscience. At the end, I will also give you an idea how our Codex technology is also used for different kind of applications. And for those of you that are not so familiar with Codex, because since it's a Phenotyping user group meeting, you might not be so familiar with Codex. I give you this little overview here. So on the right-hand side is the technology you are probably all familiar with. We see our Polaris scanner on the right-hand side. We see our reagents here, which are the Opal reagents. And you might be also familiar with the right image on the bottom side, which is a phenotyping picture of our INSERM analysis software. On the left-hand side, you see our Codex technology. This is our other platform that enables you to go up to 40 markers. It's a little bit different in technology, also in terms of reagents, because what we use are antibodies that are linked to a barcode, and those barcodes then are recognized by a specific reporter. And in a sequential way, we can fully automated visualize up to 40 biomarkers with the Codex system. Now, starting off with some publications, and this is a publication from March this year, and I would like to share this publication or show you this publication because I think it gives you a nice idea how you can, even with our old system, the vector-free system, fully automate a full workflow using a lot of panels here. On the right-hand side, you see that there were 5 6-plex panels used, and probably you are familiar with all the markers that are in there. Most of them are immune markers, and you see there's this quite diverse combination of these different markers here, and all of these markers were then used in a 7-color staining to visualize and to analyze. So the OPAL technology was used on the multiplex stainer platform BondRx from Leica, and after that it was scanned with a vector-free platform. All the pre-scans were then annotated and multispectrally imaged again with the vector-free platform. After that, there was a cell segmentation, so image analysis going on. And after that, using the INSERM software to do cell segmentation, they used MATLAB for continuous analysis here for looking for double and triple positivity of cells. And this also shows you that the OPAL staining is also capable to mark several markers on the same cell compartment and to analyze it properly. This is just an example here. So you see on the left side the composite images, and on the right side how phenomaps are created. And finally, they used this fully automated workflow then to look for cell densities from the different markers. Now I jump to the next application here. You see, this is a publication where people try to vaccinate and to analyze in prostate cancer if it could get an increase of T cells after the vaccination here. So what they did was they used the ProstVac and analyzed finally different immune markers to see if they find an increase or a decrease around the tumor in the invasive margin or in the prostate gland itself. So here you see that they could scan the whole tissue section. They could annotate the core of the tumor, the invasive margin, and also the benign glands here. And what they found out after vaccination was that they got a slight increase in CD4 cells, especially in the invasive margin, but also they saw increase of CD8 cells in the benign glands. And this was not an immense increase, but it was a 1.5 fold increase of CD4 in the invasive margin and in the gland for CD8. So that shows that you can vaccinate and also push the T cell response for tumors. And that was also shown with our technology here. This is actually a pretty old paper here, but I think what's really interesting here, it's about tumor microenvironment, but it's on pancreatic cancer, which is a very, very difficult cancer to work on. And what you might see here is that they used a lot of markers, so it's an 8-color staining, and They did an analysis where they used our INSERM software, and in addition to that, also our Phenopter scripts that are R-based to analyze distances to tumor cells. And not only that they measured the distance of certain T cells to the tumor cells, but they also measured if within this distance there's extracellular matrix involved, which means if the extracellular matrix has any impact of immune cells moving or invading into the tumor microenvironment. And here they could show that the infiltration of immune cells has nothing to do with desmoplasia, which means the extracellular matrix has not such a big impact on the invasion as people suggested before. This is also an interesting study because it was not only done on human samples, but also on mouse samples using really a big mouse panel here. And also here they used the vector 3. Now, this is an example where people used, or the group in Frankfurt of Andreas Weigert and Bernhard Brüne, they worked on inflammation in a mouse model. And although you don't have such a high amount of markers here, the mouse skin is quite difficult because it has a high, there's a very strong autofluorescence in there. And the multispectral technology is able to unmix and remove the autofluorescence, which gives you then much more reliable data here. On the bottom left, you see an example of skin tissue. This is actually not from the publication, but it illustrates nicely how the autofluorescence was removed from the skin, as you see then on the right side of the left panel. Where we are very proud of is also the transplantation field we are moving now in more and more. And this is also a pretty recent publication from April this year, which made it then to the cover of the American Journal of Transplantation. This is about kidney allograft rejection, and this work was done in Paris. And also here, interestingly, they used quite a big panel. So they used 6-color opal stain and also vector imaging to analyze this. So the question here was about the antibody-mediated rejections or T-cell mediated rejections. And as a most apparent marker here, they looked for NK cells and looked whether these NK cells are more in blood vessels, more outside, and had to look for the localization of these NK markers. Interesting here as well is that they used the NK46 as a marker and not as commonly used the NK56. So they found that this NK46 is much more specific, and the outcome was also quite important here, and that's why I think it made it really to a nice journal here. And they could now differ or look at the compartmentalization between TCMR and ABMR, and they could show that NK cells co-localize with the T lymphocytes and macrophages and appear as a cell type engaged in renal rejection. Another transplant study that also came out this year, a little bit later in the year, was done with the help of our department. And this is about cardiac allograft biopsies. And if you look into the image here, you see the classical workflow is the conventional, that you look at bright field images where you look with H&E, and actually, you just look at the amount of immune cells, and if you see a low amount, then it's a low grade. If you see a lot, then it's a high grade, and this is how you classify, and then you hope that you can make a prediction if a transplant will be rejected in the near future or not. Now, in this work, they did now a different setup. They used multiplexing technology and looked in fluorescence now and looked if this low grade and high grade are really or could be evaluated a little bit more precisely. And this is an example here. So on the left-hand side, you see the H&E stain, and then you see the multiplexing on top. And in the upper row, you see these are low grade determined, and they indeed showed clinical silence. But in the second row, they were also determined as low grade, but they were actually clinical evident. That means a rejection took place. And here you see an example of high grade, but high grade with clinical silence, and a high grade with clinical evidence. So you see, obviously it's not sufficient just to look at the T cell or at the immune cell. So you have to look a little bit more in detail, and this was done with a multiplexing here, looking at CD8, CD68, PD-L1 and FOXP3, which is more or less a classical immune-oncology panel. And PD-L1, which is more or less the most common used marker, is way not efficient. So especially on the left-hand side, you see that in the low histology grade, you see a very high PD-L1, which was expected, but also in the high histologic grade, you see that PD-L1 can be really high, and vice versa. So you need a subset of different cell types, of different immune cell types, or maybe even other cell types, to predict better which are which cases have a rejection soon, which have it in a few months, or maybe even in a few years. And this can be done now much better with a multiplexing. However, this is just a proof of concept study. We will hope that this can go further. Now, jumping to neurosciences, we have also done images on brain tissue here using a white panel here. And also, this is probably also the main part of this story. The autofluorescence in brain tissue can be also awful and really give you a false outcome of your data if you are not able to remove it properly from your images to get a good analysis in your software later. And that could look like this here. You see, when we remove the autofluorescence, you get so much more valuable data out, and the signal to noise ratio is so much better than before. That's what you see actually in the table on the left down side. Now, I also mentioned the Codex platform, and you see the Codex platform is not specific for a certain application. You can use the Codex platform in all tissue types you want. You see even in PBMCs, we use the Codex platform, and there is space for many more applications here. And I just want to give you one example. This is a FFPE section of a human breast cancer. And in this, we used a panel of 36 markers here to screen this through and to look what's really going on. And maybe some of the markers were not completely clear what they are doing. Anyways, we have the possibility on the Codex to use so many markers. and to see if they could have a relevance. And indeed, here in this example, we found something, and you see on the top part, this is a T-SNE plot. A T-SNE plot is already like a little cluster here where you can visualize a little bit how the expression is going on. And what the scientists together with our team found out that there are indeed some interesting outcomes here, and those outcomes were only possible to find by using such a high marker panel here. And finally, they found a certain cell group in the lumen of the cells here, which were KER8 and KER14 cells. And this was something that was already suggested, but could not be shown as such without this technology. With this, I would like to end here and give you a summary again. I showed you how you can use this workflow here from staining, imaging to data analysis, even on a Vectra-3 platform. Still, you saw a wide diversity of different applications that we can use. We can use the codec system up to 40 markers for discovery purposes. We can do a nice visualization of data with any platform using Phenotyping or codecs. And we can give you with our technology a big throughput with high dimensional data here, whole slide scan, and on top autofluorescence removal on the Phenotyping side. And with this, I would like to thank you, and looking forward to our next speakers.
Fantastic. Thank you very much, Bjorn. Thank you for your persistence with the sound, and thank you to our audience for your patience there with us. I tell you what, I particularly found very interesting the recent proof of concept study on the heart allograft rejection. Yeah, a really nice study helping us sort of diagnose future clinical rejection. So yeah, a very valuable practical utilization of the technology. So thank you very much. So our next speaker today is Dr. Carlos de Andrea, an MD pathologist at the University of Navarra. Carlos leads the ImmunoPath lab, which is focused on the development and validation of multiplex immunofluorescence panels for immuno-oncology research and automated digital analysis. Carlos, thank you very much for joining us today. If you could share your screen, that would be fantastic.
Thank you very much, Akoya, for inviting me. And during this talk, we'd like to tell you some of our projects using the Vectra Polaris. And as you said, the main focus in our lab is in immuno-oncology. But we have used all the advantages of the Vectra Polaris platform to also study non-tumor diseases as tuberculosis. As tuberculosis is an infectious disease caused by the microorganism, the Mycobacterium tuberculosis. And according to the WHO in 2018, an estimated 10 million people have developed active TB disease, and several of them died. As we know already, histologically, TB is characterized by the formation of granulomas, and infection begins when the microbacterium enters the lung through inhalant and reaches the alveolar spaces. Then the resident macrophages try to phagocytose it, and if fails to eliminate the bacteria, the mycobacterium invades the lung interstitial tissue, and then it will start an immune response. This immune response is characterized by the recruitment of some immune cells, including CD8 and CD4 cells and CD20 cells that will begin the formation of the granulomas. Therefore, the granulomas is a highly organized immune cell aggregate that originates in response to the infection of, in the case of tuberculosis, of the mycobacterium. And in these processes, the macrophage plays an essential role in TB granuloma formation. They also harbor the bacteria and has the power to destroy it. And once the mycobacterium gets in contact with the macrophages, they start to differentiate into epithelioid macrophages and multinucleated giant cells. And then our aim here was to try to study the complex TB microenvironment. And for that, we developed 2 multiplex panels targeting some of the most common immune cells. We have used CD11b as a myeloid marker, CD68 to stain macrophages, and then to characterize the T cell population, we used CD3, CD4, and CD8, and the B cell CD20. And additionally, in one complementary panel, we used FOXP3 to characterize the T regulatory cells and some activation markers as CD137 and PD1. And what we have done is to study all this infiltrate in here that surrounds the necrosis that is located inside the granuloma. Is the first step of our research was to develop a multiplex panel. And in here I can go briefly with you through all the validation steps that we have implemented here. Basically, we have 2 layers of validation. The first one is the run-to-run validation, where we try to replicate the same results doing the experiment in a different way, in a different day. And then the second layer of validation is the singleplex versus multiplex validation. As you can appreciate in here, you have an image with the multiplex, with all the markers, CD20, CD3, CD8, CD68, and CD11b. And in here, the comparison between multiplex and singleplex. And also, we perform some digital pathology analysis to quantify all these cells, and the quantification of that, and then we quantify until having similar cell counts in comparing singleplex to multiplex. And this is how we use to implement and set up our multiplex panel, and also the concentration of the antibodies. As you can see in here, most of the time during the multiplex implementation, we always have more signal. We have always more counts than the singleplex. This is expected, as the way that the OPAL works is a several rounds of antigen retrieval, and then you expect always to have more signals or more epitopes or more antigen in the tissue. This is another layer of validation, where you have in here our conventional immunohistochemistry as being our gold standard method, and then we compare the gold standard results with the multiplex results until we have similar staining pattern and staining this distribution. And once we have set up the panels, we performed the experiment in 14 TB clinically cases from patients with TB. And as you can appreciate in here, we have the H&E of these granulomas showing the central necrosis in here and the crown of immune cells surrounding the necrotic center. And in here you can appreciate all the markers used in this panel. And what we have done was to evaluate the immune cell infiltrate located at the periphery of the granuloma. And for image analysis, we have used the INSERM software to generate multi-TIFF image, especially to subtract the background of the slide. And then for cell phenotype, we have used QPath software. The big advantage of this software is that you can perform whole image analysis, and also this is a really fast software. Then the first step for the digital pathology analysis is the cell segmentation. As we know already, the cell segmentation is based on the detection of the DAPI of the nucleus, and then we have an algorithm to expand the nucleus, and then we have the cytoplasmic region. And then doing so, you can later on develop some algorithms for cell classification. And in here, you can already appreciate the power of this algorithm and recognizing the phenotypes that we are interested in. And in here now showing you some of the results. And in here you have two examples of granulomas, where you have the periphery in there, the area rich in immune cells, and the central area of necrosis. As you can appreciate here, you have one granuloma and another one. The scenario is completely different. In this one is full of immune cells, and that one has less immune cell. And as you can appreciate in here in this graph, out of the 14 granulomas, basically each one has its own micro environment. And also trying to and once you have all the cell densities, you can run some correlation analysis and see what cell will correlate with the other cell. One interesting thing that we found was out of 14 granulomas, we used the Zil-Nielsen staining to visualize the mycobacterium and to classify if the mycobacterium was present, yes or no, in the tissue that later on we stained with our multiplex panel. In doing so, interestingly, we found that in the cases that the mycobacterium was found on the tissue, had more CD8 and more macrophages. And then it does make sense that while the macrophages are in there trying to phagocytose the mycobacterium, and the CD8 cells are there trying to destroy the infected cells. Another way that we use the Vectra Polaris platform was to characterize if some of these cells are reactive, yes or no. And then we have some of the T cells are reactive, yes or no. And for that, we use the PD1 staining and the CD137 staining to characterize if there are some reactive T cells inside the granuloma. And what we found was that indeed you see some reactive cells, but they are a very small population among the CD4 cells or the CD8 cells. Another interesting feature is that all the giant multinucleated cells that I explained the formation of them in the beginning of my talk are positive for PD-1 staining. As you can nicely appreciate, here is a very nice membrane staining showing the positivity for this marker, and all the gigantic cells, the multinucleated giant cells, are negative for CTLA-4. And then just concluding my talk, well, I just wanted to show you that the multiplex panel can be used in other non-tumoral or non-cancer lesions, and is a very good technique that you can identify several markers in one single cell, and also you can study this spatial association between the immune cells. And also going to our studies, we just highlight something that we know already, that the immune cell infiltration in TB granulomas are very heterogeneous, and they may represent, and every granuloma may represent a unique immune microenvironment that maybe is dependent on the host response and the bacterial state, and of course in the bacterial load in the lesion. And I would like to end my talk. Thank you all the people in our department that helped us to develop this panel, and thank you for this opportunity.
Thank you very much, Carlos. Very much appreciated. A great presentation there. I'd say it was really appreciated you sort of sharing your methodology so openly about how you developed your panel. I think that's one of the intentions of this meeting is for us as users, as a community, to share this methodology. And it was also really cool the way you sort of highlighted, the different rounds of antigen retrieval, and how that enables more epitopes to become available. So it's a really nice feature of the Opal technology. So thank you very much for your generosity in sharing that. Again, I think there are questions to be answered at the end around that study. So thank you very much. Okay, fantastic, superb. So we'll go through the questions one by one. Virginie and myself will ask the question. So Virginie, would you like to go with the first question, please?
Yes. So there's a question to Yvonne. Yvonne, have you considered to stain, for example, Claudin 5 and link that to a higher accumulation of, for example, T cells in glioma sites, indicating that the therapy might benefit? Oops, sorry, it's moving when I read. Might benefit from a disrupted BBB.
Yeah, thank you for the question. Actually, we do know, we have been staining the vasculature vessel markers, BBB markers, and assessed the lymphocyte infiltration. We've also done permeability studies, and we know that the T cells, or cells that can enter, they need a functional BBB, so they need more normal vessels. So we know that the cells are not just invading the tumor through disrupted or leaky vessels. So they need to have a certain set of adhesion receptors that allow a sequential entry of those cells. It's the so-called multi-step model for leukocyte invasion. And of course, we are now with the multiplex approach trying to see how and where those cells are localized upon therapy, in particular in the preclinical model. But that question, yeah, it's valuable. Thank you very much for that. So we need yeah. I don't know if that answers the question.
Yes, I think you did. Thank you.
Thank you very much. Thank you. So we have a question here from Gamal, and it's the question is, excuse my ignorance, what is the name of the plot used to describe the different cell types?
I think there was a question from the early from Björn's presentation, right? I think he refers to Disney plot, and Disney plot is a plot to basically show high-dimensional data into 2 dimensions. It's similar to principal component analysis, if people are more familiar with that term. But Disney is just more sophisticated and more applicable to biological data sets, so high-plex biological data sets.
Okay, thank you. So now I think, Ivan, did you have a question sent to you for Carlos?
No, it wasn't me.
Oh, I beg you. Sorry. I beg your pardon, Yvonne. Sorry, I meant Ivan, one of the other panelists.
Yeah, I can ask it live because I wasn't able to type in.
Thank you.
Yeah. So just a question. Maybe I didn't see in the presentation. Have you considered a comparison analysis between latent state granuloma and the leaking granuloma? Just to further assess the immune cells response. I mean, if there is difference in the immune cells compartment into between the latent state granuloma and the leaking granuloma. Thanks, Carlos.
Yeah, thank you, Ivan, for the question. Oh, this is a good question. I mean, I think the real problem here is the sample that we have available, right? I mean, we all depends on the samples that we have in our archive. I mean, to do a really good study, we need ideally an animal model, right, where you can study different stages in the development and then compare the early stages with the late stages. But unfortunately, with human samples, at least in our hospital, is not feasible to do that.
Thanks, Carlos.
Thank you.
Thank you. We have a general question to all speakers. Have anyone ever tried Q scoring with your marker using INSERM?
Well, we haven't tried.
Okay.
And no, I didn't either.
We also didn't.
Okay. Thank you very much.
No problem. So I think that's one that we can look into offline and get back to the question on that one. Okay, fantastic. Thank you. So the next question is from Francesca Werner. Daryl, is anybody using the Vectra-3 and QPath or Halo to analyze, especially for a 2-step tissue segmentation, e.g. tumor center or tumor margin? Tumor area or lymphocytic area.
Yeah, I mean, here we are using a combination of the 2 softwares, the INSERM and QPath. What I think is the QPath is easier to annotate the sample. I mean, to establish what is your, for example, the margin of the infiltration margin of the tumor, and what is the central area. You can easily annotate and has a very nice tools that can help you to do that. With Halo, yeah, we don't have experience with the software.
Thank you. Okay, Virginie, do you want to go to the next question?
Yes. I think this one was for Yvonne. Could you please compare staining quality between Bond RX from Leica and LabSat Luna4?
So we have been using the LabSat for all the images I showed, but there's no quality issue in our hands. So the Leica Bond has been used by another group or collaborators within the Frankfurt Cancer Center, and they do report fine staining. So it's just the amount of slides you want to process, but we're establishing, and also we have two lab sets at hand, so we can do staining simultaneously. And also for our initial purposes, we are fine with that because it also needs some time to analyze the slides. So quality issue should not be there. It's just differences with regard to volumes of reagents you need, and yeah, but high throughput, of course, you need like a bond.
Indeed. Thank you. Thank you for your reply.
Thank you. So I'm the next question. I'm just trying to follow the order, but I may have it wrong here. This is to Bjorn. I think it was around about your presentation. How many markers did you use to identify a population? And that's from Carolina.
So this is also going back to the question as well that Rob answered earlier on. This is actually the whole panel that we used in the breast cancer. And you see now the Tisney plot can be used quite efficiently here. You see all the markers, and you see these rainbow colors going from blue, where you have no expression or very little expression in cells, to up to red, where there's very high expression in a certain population. And when we look now on this little island of the Tisney plot of cells, you see on the left-hand side that there's keratin 17, 5, and 14, which are strongly expressed in this island there, and also TP63 underneath. And on the other hand, you see keratin 19, 18, and 8 on the other side here, which also build a cluster. And this allows you now to really identify specific cell types here. I think this also answers your question. You saw on the Tisney plot, there were about 4 markers that identified these specific cell types. And we could now see you had these luminal cells, luminal cells with keratin 8 and 14 positive, and the basal cells, and they were all in these luminas there.
And there's another one, very general. Would it be possible to determine stem cells and assess their migration from regenerative medicine purposes? For regenerative medicine purposes?
Yeah, if you have markers to assess, for sure.
Yes.
That's possible.
And I think he wants to know the markers, actually, to determine stem cells.
I don't know offhand, but there is cancer stem cell, brain cancer stem cells described. I don't know it off my heart.
Okay.
Be able to discriminate.
Okay, thank you, Evan. Yeah, I think that's it. Also, Evan, I had a question for you in terms of, when you went from flow cytometry to Phenoptics.
Mm-hmm.
Have you done any comparative studies going from flow to tissue cell expression analysis? In terms of phenotyping?
Yeah, we're in the process, actually. We're reanalyzing our samples, and we have other ongoing studies, but I'm expecting similar results. I mean, you need both the information of numbers in the localization, and with Phenoptics you can get this information. But we haven't reanalyzed our samples, but yeah.
Okay.
Fantastic. So I think we've gone through the majority of questions. There was also some really positive comments to the speakers, about how fantastic, I mean, I think verbatim, how fantastic the presentation.
Ben, you sound very muted.
Beg your pardon, Rob. Thank you.
Better, much better.
So yeah, in the comments and chat section, it was quite clear there was some lovely feedback for the speakers on how fantastic the presentations were. So now obviously we're at the end of the meeting, and I think we can all agree that the speakers have been incredibly generous with their time and efforts in giving us such thorough and complete discussion points. So I would again like to thank Yvonne, Carlos, Huda and Bjorn for providing the great content for this meeting. And also I'd like to thank you, our guests, for taking the time to join us today. You know, we really appreciate your engagement with the questions and your interest in the solutions as well. I would like to comment that if you have any further questions following this, that we can route your questions to the speakers, or you can direct questions to us on the email address that's highlighted on the screen there. All of the webinar series will be posted onto the Akoya Biosciences website, so you can go back and review. If there's any points that you missed and you want to follow up with us, please do. And finally, at this point, you know, if you haven't registered so far for next week's webinar, please do. We'll be focusing on research involving the super high-plex Codex platform. So Bjorn touched on that platform. The majority of the talks today were around research on the Phenoptics technology, but we'll be talking about Codex next week, which is the superplex alternative to Phenoptics. So once again, thank you to the speakers. Thank you all for joining us and have a great day.